Right, welcome to DNA Lesson 1. So we're going to be going over the structure of DNA, so how it's made, what the basic function of DNA is, and then how DNA copies itself. So if you remember in our meiosis cycle, right, we had our cell cycle here, and you had G1, S, and G2, and then you had hemat or mitosis or meiosis. Well, during the synthesis phase, DNA copies itself, and we're going to be looking at exactly how that happens today. So, moving on, first of all, traits. We want to go with our vocabulary first as normal. Traits are going to be characteristics that are inherited. So, these are things that you inherit from your parents through your DNA. And DNA itself is a nucleic acid, so taking this back to macromolecules, it's a nucleic acid that stores and transmits genetic information. So this is how the traits are passed on. Heritable means that it has the ability to be passed on from one generation to the next. And the central dogma here, so when we ever talk about that, is that DNA, in addition to carrying your information for your traits, like in addition to determining what color hair you have, your DNA also has information for coding for proteins, so for the productions of proteins. And we're going to be talking about that in lesson 2 and so forth. So you can see here we have our double helix of DNA, and I'm going to trace the shape of it here, that is connected together by nucleotides. So we're going to go over that in a couple minutes. Genetics in general is the study of hereditary patterns and variations in organisms. This is what we just finished covering in our last unit. So when we were covering things like Punnett squares and codominance and incomplete dominance, things like that, those are hereditary patterns and variations. So there were several people that their work accumulated into the discovery of DNA. One of the first ones we're going to talk about is Griffith. He basically experimented on mice, and he observed some harmless strains of bacteria that could change into harmful strains. And this process he called transformation. So basically what he did is he looked, when he injected the live bacteria into the mouse died. When he put in this live R bacteria, mouse lived. So this one was harmful and this one was safe. So harmful and safe. Now, he took this S bacteria, the harmful bacteria, killed it with heat and injected it into the mouse. The mouse was good. He lived. But when he took the S bacteria that had been killed with heat with the R bacteria that should have been safe, so these are both should be safe, the mouse still died. And this is because the bacteria, he found that this dead bacteria could still put DNA into this live bacteria and cause this live bacteria to turn harmful. So Avery then looked at that transforming principle. He looked at the work that Griffith did and saw that maybe it's actually DNA that's doing that. So Griffith just knew that the combination of these two bacteria would interact with each other. So what Avery did is he isolated and purified Griffith's principle. So he took the science that Griffith did and expanded on it, which is 100% the core values of scientific research. He performed 3 tests on the transforming principle. Qualitative, meaning yes or no answers, we're not looking at actual numbers, we're looking at values, showed that DNA was present. Chemical tests showed that the chemical of makeup matched DNA, so we know that it was a nucleic acid. An enzyme test showed that only the enzymes that would degrade DNA actually stop transformation. So he proved that that transformation that Griffith showed with the mice happened because of DNA. So Avery's conclusion, his principle, is that DNA is the nucleic acid that stores and transmits the genetic information from one generation to the next. Finally, we have the Hershey and Chase experiments. So what they did here is they took a bacteriophage, looks like this, and it looks like that in real life. It's kind of crazy. And was able to inject the DNA from this bacteriophage into a bacteria, right? And when that bacteria, when that bacteriophage, when the DNA was radioactive, then we got radioactive DNA inside the bacteria. But when instead the radioactivity was on the outside, so when they were on proteins and then the DNA was injected, there's no radioactivity on the DNA inside the bacteria, which it seems pretty obvious to us now. But basically what he was doing is he was trying to figure out, is it proteins that are being transferred or is it DNA that's being transferred? And he concluded that the genetic material in bacteria was DNA, not protein. For a long time, we thought the genetic material was actually protein. So that was basically how we discovered DNA and what DNA does. But now let's get into the meat and potatoes of what DNA is. So in your notes packet for lesson 1, you should be on exploration 2, the structure of DNA. So we're going to be looking at nucleotides. Do you remember back in our macromolecules lesson, nucleic acids, the monomer is nucleotides. Each nucleotide, so that monomer, has 3 parts. It has a phosphate sugar. Excuse me, I circled the wrong thing. It has that's the phosphate group. It has a phosphate sugar right here. And so we know it's a sugar because of that hexagon shape or that pentagon shape. It has a phosphate group, which is up here. And these two together make up the backbone of the DNA. Then it connects to a nitrogen-containing base. And this is what we will be spending most of our time talking about. And so this is the basic shape of a nucleotide. I'm going to erase all my images here so you can see this. This shape you will need to recognize as a nucleotide. And remember, you have your phosphate group here, your sugar as the pentagon here, and the nitrogen-containing base. So that can have a couple different shapes. So this was the image from that first slide here. You can see there's our sugar and phosphate over here in the backbone, and you have other ones over here as well. And they connect to this nitrogen base, which is out here. And the nitrogen bases are in the center, and you can see some of them are just one shape, and some of them are two shape, and those are going to be our two differences. The different nucleotides have different names. There are actually 4 kinds, and these 4 kinds are divided into 2 classes. So you have your purines, which is that last line on your notes, and this is adenine and guanine, so abbreviated as A and G. Then you have your pyrimidines, which are cytosine and thymines, which are abbreviated C and T. So your 4 letters of your DNA are A, T, C, and G. So imagine if we had an alphabet that's only made of 4 letters. And how DNA works is these 4 letters are put into combinations of 3 letters. So maybe like ATC or CGA, and there's a bunch of different combinations of these 3 letters. Those are like our words. Then these words are put together in sentences, and that's how your DNA codes. But the whole DNA alphabet is only 4 letters here, A, T, C, and G. Now we'll be introducing a 5th letter when we talk about RNA in our next lesson. So here's our nucleotides again. You can see our purines are shaped a little bit differently than our pyrimidines, right? And so so you have your purines and your pyrimidines there. Now, in order to determine DNA structure, here we go. So here's where we're talking about the shape and the structural formula here. So when you're looking at this slide, you can really look at the differences between A, T, C, and G. You will not need to memorize all these letters. You will need to know that A and G are purines and T and C are pyrimidines. We have to determine, now we determined that DNA is what transfers genetic material. Those were the first studies. Then we wanted to know, what does DNA look like? How is it built? So we discovered that it's built of these nucleotides. But several different scientists, Chargaff, Watson and Crick, and Rosalind Franklin, showed that how DNA was built. So Watson and Crick determined that there was a three-dimensional structure of DNA, and they did this by building some models. And so this was their first, this is Watson and Crick here, and this was their first big DNA model. Their discovery was not out of thin air. So they built on the work of Rosalind Franklin, and Erwin Chargaff. So Franklin, who did not get credit for her research for a very long time because she was a woman, but now she receives the full due credit. She suggested that DNA was a double helix, which means it's this shape here, right? And that twisted shape that we see in DNA. And of even width, meaning no matter where you measured the DNA at, it was the same width apart. Chargaff figured out that with those nucleotides and how they pair up, is that A, which remember is a purine, always pairs with T. So these always go together, which is a pyrimidine, and that C always pairs with G. So he discovered that there. So this was Rosalind Franklin, and this was one of her, that's Rosalind Franklin there. And then this is one of her X-ray images. So she did a lot of X-ray images of DNA and was able to figure out that when you look top down, it looks like an X because of that double helix shape. Okay, so Chargaff's rules. Remember we said that A always pairs with T, C always pairs with G, and this keeps them that even shape. You see how these are two things wide. If we paired A with G, the DNA would be wider in some spots than others. So we always want A paired with T and G paired with C. And I'm going to reiterate that again. Nucleotides always pair in the same way. And go through our slide here. See that. Okay. Backbone of DNA. So the backbone here, which of course this would be twisted up when we start talking about it, these are connected by covalent bonds. I'll learn more about in chemistry next year. The bonds themselves, the nucleotides are connected together by hydrogen bonds. And that's because the hydrogen bonds need to be split up and be connected whenever DNA replicates. So, as an overview of the whole thing here, and this kind of fills in the sentence of your current DNA model in your notes. So there's a sentence with some blanks in it. The current model represents DNA nucleotides of a single strand are joined together by hydrogen bonds that connect the nitrogen base of one nucleotide to the nitrogen base of another, so A to T or C to G. The alternating sugars and phosphates form a chain or a sugar phosphate backbone. So that's what the sides of the DNA is held together with. The DNA double helix is held together by hydrogen bonds between bases in the middle. So this is the overall structure of DNA described, and you'll be doing some activities where you're drawing this out as well. This is basically that same exact sentence, but put together into visual images. So you see we have our covalent bonds here along the side, and that's going to be our backbone. So these are the backbones, and then these are the hydrogen bonds connecting the nucleic bases. So I like to think of DNA as kind of like a twisty ladder, right? A ladder at a fun house. So the sides of the ladder are here, and that's going to be the backbone. Whereas the rungs are what you're actually climbing, those are the hydrogen bonds of the nitrogen bases. Okay? And then you take that ladder and you twist it into this shape here. So you can see C and G pair together, A and T pair together. You have your backbone here along the back, and then your nitrogen bases in the center. So DNA replication. So we're now on to the exploration 3 there. And this has a very short part of notes, but you're going to want to be drawing some of this out. This is the process by which DNA is copied. So like we talked about, it happens during the S phase, and it's carried out by proteins and enzymes. So proteins and enzymes are what are doing DNA replication. So the DNA serves as a template, and then 2 new DNA strands are made. So enzymes and other proteins, like I said, do the actual work of replication. First of all, they unzip the double helix, and then free-floating nucleotides, which you get from your food and your body can make as well, those form hydrogen bonds with the template strand. So you can see that the DNA unzips right along the center here, and that these new nucleotides are coming in and attaching to the template strand. DNA polymerase, these are enzymes that form bonds between nucleotides during replication. Basically what this does is it proofreads the strand and makes sure that each molecule is a perfect copy of the original. This is involved with our G2 checkpoint. Remember how I said back way during macromolecules, if a cell doesn't pass this checkpoint, the cell dies. So that's DNA polymerase. They're, TSA people that are sitting there and saying, yes, you can go, or no, you cannot. So that enzyme is what does it. These enzymes also bond the nucleotides together to form a double helix. So they go back and basically proofread everything, bond it all together. They form the new covalent bonds between the nucleotides of the new strand. So your original strand here is in blue. It is being unzipped. The new nucleotides are coming in to attach and then forming a whole new strand. So this orange strand is a new strand of DNA. So if this process continues along the entire strand of DNA, you go from having one strand to 2 strands that are identical. The 2 new molecules of DNA are formed. Each one has an original strand and a newly formed strand. I'm going to repeat that because that is extremely important. So if you're a little zoned out right now, come back to our lesson, because you do need to know that 2 new molecules of DNA are formed, but each of those new molecules have an original strand and a newly formed strand. It's kind of like having a veteran and a newbie. Or if you're into Star Wars, a master and an apprentice. You always have an original strand and a newly formed strand. We call this the semi-conservative model because you're conserving one strand, but it's only one strand, so it's still semi-conservative. So you can see we have 2 new molecules of DNA. Each one has an original strand, each one has a new strand, and this is called the semi-conservative model. It's almost like this is a question on your quiz or test and definitely in your EOC. So the semi-conservative model says that you have an original strand and a new strand in each new molecule of DNA. So replication is extremely fast and extremely accurate. Your body does this every time you do mitosis. You have trillions of cells that are carrying this out all the time. It starts at many points along the DNA, and this is for eukaryotic cells. So we're talking about your own body cells here. So you have this long strand of DNA here. Replication can start here and here and here at the same time. And they're going to make a better drawing of that. So you can see this is where we started replication. We started replication here and here. And then the replication works outwards until it meets up with the other strands. This makes it go a lot faster than starting at one end and working all the way to the other. That would take a lot longer. So you can see here we have another picture of it. We started out, we brought in some new nucleotides, and we added a new strand onto our original strand. So that's the process, that's the structure of DNA, how we discovered DNA, and the process of DNA replication. You have a worksheet that we're working on today, as well as a DNA foldable that we'll be working on as well. And one or the other is one day or the other. So make sure you're catching up with those assignments, checking Canvas to rewatch the videos, and making sure you're sticking up with all your assignments. Right, so that's the end of the slide. If you need to rewatch it again, it is on campus.